Yes, sir.
So should we start or no?
We'll wait another 2 minutes and then we can start.
Okay, thank you. I'll just come back.
So in the meantime, I just wanted to ask, did you decide on the mode of assessment?
Yeah, so we'll have a meeting today, and then by tomorrow I'll let you know. Is that okay?
Okay.
Yeah. So by tomorrow, before tomorrow 11, we'll let you know.
Also, what are the days that you want to take class?
In this week, because the materials prof is also asking for he was asking for some free time.
Oh, so that tomorrow, day after tomorrow, and the day after. So we'll be done by Thursday. Well, actually we can try to get done by Wednesday, and if it's not done, then we can go to Thursday.
Okay, so the slot. Which slots do you want to take these classes? Because
The
he was
usual
asking
time.
for some.
The usual time, right? So the 11 to 12.
Okay.
Yeah. So we don't have any other extra classes. So today is the last extra one, like which I'm making up for the ones that we missed.
Okay. Thank you.
All right.
Hello, sir.
Yeah.
So before the exam, could you, outline the chapters that you have covered from the reference book so that we could, go through those chapters only while referring to the book?
Yes, I can definitely do that. So tomorrow we'll let you know that how the test will be done, and also let you know the chapters and kind of as much indication I can give. Right.
Okay.
Yeah. Okay, so we'll start for today and other things to do. Yeah. So today's lecture would be about the auditory system and the somatosensory system. And so, when we hear sounds through we perceive sounds through our auditory system. So the idea of that, the high pitch, high frequency versus high intensity. So when high frequency means that it's higher pitch or it's much shriller, and high intensity means it's louder. And of course, the sound travels through air, and since the velocity is constant, if the y-axis is the air pressure and then the distance, so if it's if the sound it comes in a higher frequency, then that's what you perceive as a higher pitch, as I said, and also if the intensity is higher, that is the amplitude is higher with time or distance, then you perceive it as basically louder sound. And so we have of course a range that we can hear. So then higher than that would be ultrasonic, and below that would be subsonic. And different animals vocalize at different frequencies. Can hear at has a hearing range of different frequencies. So like humans, it's somewhere between 20 hertz to 20 kilohertz, and below that would be the subsonic the subsonic sounds and subsonic frequencies, and the supersonic ultrasonic frequencies. And these are a comparison of different laboratory animals compared to humans. And you can see that mouse has somewhere, between 2 to like 65 or 70 kilohertz versus something cat has a much wider range, and humans and macaques or monkeys have more similar range of kind of range of hearing. And in the laboratory, we can record vocalization, and it comes kind of this kind of histograms where the frequency are mapped with time. And so there are various ways to measure them, like various microphones and then you can kind of separate out. So you can place these microphones in the wild, and then you can separate out the different kind of depending on the frequencies, what kind of animals are making these sounds. And we perceive these sounds through, of course, the ear. And so you basically have the outer ear, the middle ear, and the inner ear. So outer ear is the pinna where the sounds are coming in and through the auditory canal. And then you have the eardrum or the tympanic membrane. So here the sound waves impinge on the tympanic membrane. And then from the tympanic membrane, the pressure is basically transduced through these small bones called ossicles into the oval window. So there are three main structures here. So you have the tympanic membrane, and then you have the middle ear, the bones, and then it goes on to like the waves basically transmitted by them to the oval window, what is called in this device known as the cochlea. And the cochlea, the sound goes through and eventually it's made into action potential, which goes to the auditory nerve and eventually to the brain. So that's a basic structure, and so this area, the cochlear region is filled with fluid, and so this device or this kind of paraphernalia, if you may call it, it regulates not just the auditory system for the hearing, also it controls the vestibular system, that is for balance. And we'll focus here on the auditory system. So as I said, something that you should remember, the sound waves move the tympanic membrane or vibrate the tympanic membrane. Then the tympanic membrane it moves the ossicles, and the ossicles then would move the oval window. So there are three things here basically, and the oval window movement is sensed with the cochlea, and the arrangement or the cells that are in the cochlea and the fluid, that vibration is eventually made into action potential in the auditory vestibular nerve. And then we basically will talk about this process in short, how this happens and what are the different components of it. And how does the auditory information go to the brain or the cortex? And it's very similar to the visual circuits or the visual pathway that we talked about in the last class. Just one second. Yeah, in the last class, so you have the auditory receptors in the cochlea, then goes to the brainstem, it goes to the medial geniculate nucleus, then goes to the cortex. Very similar as this the visual system, where the photoreceptors that goes to the retinal neurons, with the lateral geniculate nucleus and the visual cortex. So you don't have to go through the details of it, but the idea being that they are very similar in how the pathways, how they eventually end up in the cortex and give us the perception of seeing or hearing. There are many more steps in the auditory, because there are other nuclei that kind of intermediates this, but this is the general idea of information transmission. And then the middle ear this is kind of a really cool structure, because you have three very tiny bones that is kind of amplifying the signal that is coming on the tympanic membrane. So what happens here is that the cochlea is basically filled with fluid, and because this is much smaller than this, the oval window is much smaller than this, the pressure that gets on this is way less than the pressure on. So if the pressure that is given by this stapes bone, the small bone on the oval window, is much higher than what is kind of impinged here. So that's how the signal is amplified. And so what happens is, when there is the sound wave hits the tympanic membrane, it pushes this inside, and that would push the malleus outside. And the step incus, since it's connected to this, it will also move outside, and which in turn will push, because this is moving like a lever, because this is moving outwards. This will move this thing inside, and of course this happens at much faster. It's happening, as the sound waves coming in, and it's going on vibrating, and that is eventually causing the sound information to be transmitted to our central nervous system and giving us the perception of what we are hearing. And if you really think about what we can, like how the acuity of our auditory system, then you can imagine that how much kind of mechanics or what would be the range of dynamics that this bones vibrate in, so that it can transmit such complex information that we get from different auditory stimuli. And if you make the cochlea is basically this structure, kind of snail-like structure. So the word comes from snail, what is in snail in Greek. And if you make a cross section of this is how it looks like. There are 3 chambers, of which is called the scalars, and there is this membrane called the basilar membrane, and the scalars itself is are separated by Reissner's membrane, and there is this organ of corti that sits on the basilar membrane. So what happens is when the basilar membrane, when the cochlea is sitting inside and is filled with fluids, and when the oval window is vibrated, this basilar membrane also vibrates, and the organ of corti, there are cells here called hair cells that are bent, and this structure is innervated by the auditory nerve. So when it's bending, it's also mechanically stimulating the ends of the auditory nerve, and that's what causing the action potential and sending the information to the brain. And so just to remember that there are 3 parallel chambers, and just know the major structures and the membranes, and the organ of corti contains the auditory receptors, and it sits on this basilar membrane, which basically vibrates due to what happens at the oval window. And the organ of corti is covered by this membrane called the tectorial membrane. So this is the organ of corti. This is where the sound transaction happens. And so you have the hair cells, which are basically epithelial cells, so those are not neurons by themselves. And there's stereocilia, and whenever this basilar membrane is moving, this is also kind of hitting against this tectorial membrane and vibrating. And this vibration is transmitted through the auditory nerve. And the auditory nerve there's the spiral ganglion cells, which are the actual neurons. The cell bodies are within the nerve itself, and it's innervating here. One side is going here and sensing, and the other side would go to the brainstem and transmit the information. And so all of us know about cochlear implants or the hearing, or the so-called hearing aids. So what happens here is you have outside the brain there is a microphone, and that receives the information, and eventually it is wired to the cochlea. So wired to the cochlea and actually gets into the auditory nerve. So what happens is it's an artificial stimulation, so you need to train on it. So what happens is it can actually kind of emulate what would happen naturally through the ossicles, through the bones here. And what it does is it directly stimulates the auditory nerve in certain patterns. So as time has gone, this has become more improved and more sophisticated, and it can almost be like natural stimulation. And what it's doing is it basically showing kind of sending very low frequency, low amplitude electrical signals, electrical stimuli, and the micro-stimulating the nerves themselves in a pattern that would kind of reflect the sound waves that this microphone is receiving. And as I said, that it has become really improved. It has developed a lot. So now it can almost you don't need to train on it that long anymore, and within days you will learn how to hear. So as long as the auditory nerve is not damaged, this would work, and you can almost like perceive like a normal like a normal human being would, or a person without any hearing impairment would. And so as I said, that eventually the information has to end up in the cortex so that we can perceive what we're hearing. So what happens is you have the spiral ganglion that is innervating the hair cells, and from the spiral ganglion information goes to the ventral cochlear nucleus. It goes to the region called the superior olive and inferior collicus. But you don't have to know all these details. The point is that the idea is that either in any of the sensory system, be it visual, auditory, or somatosensory, there are many pathways, many nuclei that sits in the between, and they are doing their own computations, and eventually the refined information is going to the cortex, and that's where the perception is made. And how do you experiment this in the lab? So I wanted to show you some experiments that people have done in mice. So they have mapped out in the somatosensory cortex. So if you see, the cortex is a big structure, and how different kind of sounds are processed at different areas needs to be known, so that how a mixer, say you are say a same musician is playing two separate pieces of music, they only differ by a few notes or so. But if you're aware, you can actually differentiate between the two pieces of music. And how do you do that? Because your brain can distinguish and differentiate between those frequencies. And even very small changes, if you're trained on it. Training means here we have heard it many times, you understand the music, and then now you can differentiate between the two kinds of music. And how in mouse also we can test that. We can ask what is or you can do in monkeys. We can ask that how in the brain this computation is happening, what are the different regions that are perceiving that. Within the auditory cortex, what are the different nuclei perceiving? What are the correlations between these two regions? So how, if this frequency after comes up to this frequency, how would the two frequency kind of tune cortical nuclei would talk to each other? That you can determine. So in this experiment, what they did was they took a mouse, they thinned the skull, which means that so it has all the neurons are expressing calcium indicator, which are kind of a substitute or for neural activity. So they can, if it's a fluorescent indicator, we have spoken about it before. So if the fluorescence gets brighter, that means that the neurons there are firing, and you basically collecting the data. You're lighting up with the LED or with certain kind of wavelength. So using a blue light here, and then you are looking at it with a camera with a filter on it. So what you're seeing is that the fluorescence get brighter, means that the neurons are active there. And actually, realistically, it's a pretty, it sounds complicated, but it's a very simple experiment where what you have done is that the neurons are expressing this sensor. Otherwise, the mice are completely normal. And what you have done is you have thinned the skull. What means that you take a drill and you make the skull so thin that now a camera can see and the fluorescence is so bright that a camera from outside from far can see the changes or the dynamics. And the mouse is not anesthetized, the mouse is just running around and so it is head fixed, which means that the head cannot move, but otherwise the mouse can run on a treadmill or they have these balls called rotating balls, and then the mouse can it's sort of like not the newer mouse that we use with computers, but the older mouse had this tracking balls at the bottom. So it's a very much bigger version of that, and the mouse is kind of sitting and walking on it, and you can track it. And then you're playing sounds to it, and then different frequencies, and with frequencies you can see signals and different regions of the auditory cortex, and that's what's happening. So they're giving different stimulus, different frequency, kilohertz, 12 kilohertz, 30 kilohertz, and you see the patterns are very different. Then you can kind of make histograms like this, and you can see that how these different parts are kind of they correlate with each other. What you can do after this, you can silence these places and say, okay, I have silenced this place. And then if you see a perception, a behavioral difference, that means that this region probably is important for perceiving only that frequency. So these experiments will eventually help us to understand that how we distinguish between different frequency, different kind of tones, and depending on how they're played close to each other. And you can basically answer very complicated questions like that about our perception of sound. And after that, we'll move to the somatosensory system. So the somatosensory system is basically our skin and what you perceive through our skin. So we talked about the visual system, the auditory system. The visual is what we see, and so why we need to understand this? Well, basically, this is how we interact with our environment. So when we are, say, you walk into a new environment, and how do you perceive? How do you make sure that no harm comes to you? You are not harmed, or you can just go through that place very safely. So the ways you can do that is like you look around and you see, okay, everything is fine or not, if anything is threatening or not. You look for sounds that if something, say like, you are just walking on the street, you make sure there is not a car coming, a bike coming that might hit you. And then you also use your sensation of touch. So you are walking on the street or a footpath, and if you suddenly feel like there is something of there might be a hole or there might be some undulation, you make yourself careful. So I should walk carefully here because the footpath or the road you're walking on is not very smooth. So all these sensations, all these senses have to come together for you to make sure you keep yourself safe, and also not just safety and protecting yourself. Also, when you are trying to do something, say you want to pick up a pen. So what you do is you kind of look. You will look for your pen, then you use your somatosensory system. You touching the pen, so you know that okay, it's a pen, and then you kind of picking it up. And more importantly, it becomes, say you are operating in dark where you can't see anymore. So you kind of know that it must be there, and you are kind of trying to touch and look for it. So all these sensations, they complement each other. Like when one is off, the other gets that's way stronger. When somebody is visually impaired, their somatosensation and auditory system gets much stronger, and that's how they kind of overcome or compensate for what they're lacking, right? So the somatosensory system is basically the skin. So you have two kinds of skin. You have the hairy skin and the glabrous skin. So hairy skin is what covers most of our body. Glabrous skin and hairy skin, what the name means that it has hair on it. The palm of our lips, our palm, or the bottom of our foot, these are the glabrous skin where there is no hair. And so both are very sensitive. The glabrous skin is particularly more sensitive because this is how we sense, since like, when you are trying to look for something, or we are kind of, sorry. We're kind of navigating our environment. We're mostly using our hands and foot to kind of feel everything. So these are actually ultra sensitive. Even it has even more acuity than the hairy skin, which acuity here means that how close to, say, like you are taking two pens, and how you can, and if I poke on your skin, you can kind of predict how far it is. You can't say that how many centimeters far it is, but you can compare between two sets of stimuli. In a sense, I take two pins and keep 2 centimeters away versus I keep 2 pins and 5 centimeters away. If I do this consecutively, you'll be able to say the first was smaller than the later one. So you can do this kind of, your brain can do this kind of computation and can tell you that, give you the information, like kind of make you feel that information. And this all happens through the sensory receptors in the skin. So, and there are many kinds. There are Merkel cells, so there are free nerve ending, there are mycinous corpuscles, and then hair follicle receptors called the hair follicle receptors, pacinin. So they're basically different names, and they have different kind of, as you see, they're at different depths. So something that has a high pressure would be, as you can guess, would be sensed by this. If it's a low pressure and having at the very superficial layers, it's sensed by this. If you kind of bend the hair this way, that way, it's felt by the receptors here. And then they have the free nerve endings, which usually transmit pain information. So when it's hurting, it's transmitted by these neurons. So how do you, so one of the important points to kind of important experiments that are done is there are receptive field mapping, which we have spoken before. And receptor in the visual system. And so similar for auditory system, you can kind of play sound at different ranges and map the receptive field. Similarly, somatosensory system, you can map the receptor. And this is very quite important and very interesting because the skin is the biggest organ of our body, and we want to know exactly what one nerve innervates. So you can see that what you can do is you record from the nerve, and then you can do that in humans, and you can poke. So it's called microneurography. So where you basically put an electrode in your skin, and it's a very thin electrode, so you don't really hurt that much. And you can record from that, from those neurons, and then you can go poke at different, very closely poke at different areas, and then you can kind of map that each neurons would kind of innervate at particular regions of your hand. And so how do the neurons now? So say like I think I've given you an example before. So you take a pen and you slowly push against your skin. So first it doesn't hurt. What it does is you feel that some pointy object has touched your skin, then you slowly push. And what happens eventually is start hurting. And so this kind of graded response is transmitted by fibers, sort of types of fibers, so A delta, A beta, A delta, and C. So the A alpha had the biggest diameter, they can transmit the fastest because of the amount of myelin sheath, and this is a little slower, and C is the slowest. So first is the fastest response will go when you touch. So this is done by A alpha. You push more, you get A beta. A delta is still hurting, C really hurts. So, and this has different speeds. So if you see the diameter, depending on the diameter, is 80 to 120 millimeter per second, meter per second versus 0.5 to 2. So, and so these are the kind of receptors, the proprioceptors, which are kind of detecting where your hand is in the space. And then you have the mechanoreceptors in the skin, which are basically, as I said, that it the name suggests it's detecting mechanical stimuli, skin, and then you have the pain and temperature, and the again pain. So as I said, a same stimuli can kind of excite all those neurons and kind of give you the complete picture of what's happening at the level of the skin, even if you're not looking at it or nobody's telling you what's actually happening. Yeah, and then the spinal cord is basically organized in segments, and what's here is there. So you go from the sacral. So if your spinal cord looking at the bottom of sacral, that's at the bottom of your back, and then you have a lumbar, which is innervating your legs. Then you have a thoracic, it's innervating all through your chest and everywhere else. The cervical is mostly innervating your hands. And at that, and also hand and neck, and also tissues at that level. And what happens is here is that why the segmentation is so important is because the information is coming from the somatosensory system to the spinal cord, and the spinal cord has to carry it to the brain. So the information needs to be discrete at this level, so that these can be computed and sent up. And so they also go to different regions of the brain, and they have a somatography, which means that each region of the brain would only kind of connected to this region, functionally connected to this region, and then they can talk among each other, and kind of then you get the perception, okay, like my hand and leg both are hurting together, or my left hand, right hand leg are hurt, or my back is affected. That kind of spatial information you get because of this kind of segmental because if everything was connected, it would be much hard to kind of dissect one sensation from each other. So you have this kind of discrete centers in spinal cord that it receives information depending on what they're innervating. And then what happens is once it comes to spinal cord, so this is now the cross sections which we talked about before, like the cross sections and sagittal sections. So in cross sections, the sensory neurons from the skin is feeding in information to the dorsal horn of the spinal cord, and then it goes to this regions called dorsal column nuclei, and then it crosses over, which means it goes from one side of the spinal cord to the other side, and then it goes to the thalamus and then somatosensory cortex. Again, so this is a very general pathway, just like the auditory and visual. The parts are here different because it's coming all through the body. It's not directly going to the brainstem, but the nerves that innervate your face are directly going to the brainstem. So that's very different, but it generally, like all this sensory system, is kind of following a kind of a pattern where the ganglions of the sensory neurons are innervating the organs, then it's sending it to the spinal cord or the brainstem, and then it going through intermediate nuclei to the thalamus and eventually to the cortex. And so lastly, this is the last slide, and what we talk about is pain. So now this is something that's in somatosensory system, like that's the pathological importance of understanding this. So what means that what goes wrong in the somatosensory system that can kind of hurt you. So if you know about so we should talk about low threshold, high threshold here. So as I said, that when you are touching the pin on your skin and then pushing it slowly, so when the first information you got is the pin is basically touching your skin, so that is the low threshold response, low threshold stimuli. And then as you increasing the kind of intensity of the mechanical stimuli and it hurting, that's your high kind of high intensity, or that becomes a high threshold stimuli. So once it becomes high threshold, you're recruiting the C fibers, versus when you first just touch with the A betas. So depending on that, you have a high threshold stimuli and it's eventually hurting. And when it hurts, so it's not as simple as if the skin is poking, but when the skin is kind of breaking the skin, the pin is breaking the skin, it causes a lot of signals to be kind of secreted from these skin cells. And eventually what happens here, something, the sensory neurons can transmit it to the spinal cord. And when that information comes there, eventually it goes to the, brain, and it can kind of translate that information. And it's important to understand, because what happens in chronic pain conditions. So what is chronic pain? Chronic pain basically means, say, arthritis, that a lot of our parents and older folks in our families have it, or even sometimes you can develop it as young. So what happens is your kind of bone joint degrades, and then eventually damages the nerve fibers, just like here what I've shown. And what happens after that is it becomes a maladaptive, which maladaptive means that if you're just kind of walking barefoot and there is a pin and you step on it, it will hurt, and you will rub your kind of feet, and eventually it will go away. But what happens so that is kind of adaptive, it comes back to normal. In maladaptive cases where it doesn't come back to normal, what is going to happen is it's going to hurt for a while. And as in arthritis, that it kind of stays for a long time and actually never goes away. And so that is called a maladaptive situation. And that's called a maladaptive situation. And the pain will become chronic and last forever. And the nervous system remember it, and it's kind of, you will kind of recognize that this is a sustained problem and you need to go to the doctor. And the whole somatosensory system, which is designed to kind of protect you and kind of make you aware of the dangers that are lying around you, which becomes a problem now, because it's telling you there's danger all the time, and you can't do much about it because there is no real solution or real kind of drugs to cure that. So that's that. So that's basically I wanted to go through the major concepts, and then I'll list out the chapters which you want to read and the segments of it. So you don't have to read the entire chapter. And so what I would suggest to do is that if you just look at the slides, which I'll also offer the PDF, you just go through the major concepts and kind of find the major concepts in the textbook, and also just read those. And that would be that. Okay. Questions? Any questions? Okay, no? Are you sure? All right.
Sir,
Yeah.
I have a slightly not so relevant question. It's about the Kiki Booba effect.
What effect?
Maybe I'm saying this wrong. Bouba, Kiki effect. Where you
Uh,
associate shape to sound, right? What is the basis for that?
Sorry, can you repeat that? I don't know actually what you're talking about. Let me.
Bouba and Kiki.
Poupakiki. Okay, I have to look into that. I have no idea. Yeah. I can Google and see, but yeah, I have to read that out. I have no idea what is it, but I can look into it.
Okay, thank you.
Yeah. Anybody else? All right. Bye. Good.